Okay, hope everything went good. So hello, everyone. Participants are starting to roll in, so I think we can start.
Yep.
I would like to welcome everyone here at our EMN live webinar. Today, actually, is the 18th session. And for this February webinar, I'm the host. My name is Aleš. I'm a researcher in Oslo University Hospital in Norway, focusing on clinical metabolomics and lipidomics applications in the hospital. And I will be co-hosted, this webinar will be co-hosted by Loic.
Hi everyone, I'm Loic. I'm going to be your co-host today. I'm a postdoc at the Crick Institute in London, and I'm more focusing around metabolomics, but applied to the pancreatic cancer and the process of metastasis to the lung of this cancer. And I do mostly untargeted metabolomics using mass spec.
Perfect. Thanks, Loic. And I would just like to introduce you or invite you to follow us on our social media. So we are using X, LinkedIn, and Blue Sky. Also, if you have any questions further on, you can reach us by email. And please feel free to follow us or follow up with any questions you might have. But without further ado, I would like to jump to the main point of this webinar, of course, and that's the topic that we will be talking about today. And I'm so happy that we can be hosting here two excellent speakers. And it's especially quite fitting because yesterday we celebrated the International Day of Women and Girls in Science, which is amazing. So I think it's really fitting that we have two wonderful women scientists here, Dr. Judith Jans and Hannah German. So I would just like to introduce quickly the first speaker of today, and that's Dr. Judith Jans. And she is the head of Metabolic Diagnostics section at the University Medical Center in Utrecht. And she's also a laboratory specialist in clinical genetics, specializing in diagnostics of inborn error of metabolism. She is also associate professor at the Department of Genetics at the Wilhelmina Children's Hospital. And her research interests include the elucidation of novel genetic metabolic diseases, with a specific interest in developing untargeted metabolomics to aid in this process. And her recent work has focused on the introduction of untargeted metabolomics in diagnostics and enhancing the pathophysiology of rare diseases. And today we will hear two wonderful lectures in the topics of application of metabolic flux analysis in the field of inborn errors of metabolism.
Great, thanks, Aleš. And it's my pleasure to introduce the second speaker of the day, which is Hannah German, and she's a PhD student at Utrecht University in a group of Dr. Judith Jans, but also in a group of Prof. Nanda Verovendrief. Sorry if I'm butchering the name, but I think I got it almost right. And she focuses mostly on the malate aspartate shuttle, and she has studied and developed several in vitro models to increase the knowledge on malate aspartate shuttle disorders.
Thanks a lot, Loic. And I think without further ado, we can now, because all of you are eagerly waiting for the excellent presentations. I would just like to say that if you have any questions during the presentations, please drop them in the Q&A session. And we will then get to the question section at the end of both presentations. So thank you so much. I will stop sharing my screen and leave the floor to Judith.
Thank you very much, Aleš, for your kind introduction. Let me see if I can start sharing my screen. I think this is working.
It's working nicely.
Perfect.
Okay. Thank you. Well, good afternoon, and I guess good morning, perhaps, for some of you, depending on where you are right now. First of all, thank you very much for the invitation to present this webinar for the Metabolomic Society. I'm very pleased and also perhaps a little bit overwhelmed by to see that so many people are joining us live today. So thank you very much. What I would like to do and would like to discuss are the applications of metabolic flux analyses, and specifically in the context of inborn errors of metabolism. I will first introduce the topic of inborn metabolic diseases and then start off with some examples of what we can learn from metabolic flux analyses. And afterwards, indeed, as was mentioned, Hannah German will continue as one of our young investigators in our lab, and she will go more in depth and discuss part of her PhD project, where flux analyses, I think, are a very important tool. So we are here at the Wilhelmina Children's Hospital. This is part of the University Medical Center Utrecht in the Netherlands, and this is our group. We work in the Metabolic Diagnostic Lab, and of course, we need to talk then about metabolism. And I'm pretty sure that for this audience, I do not really need to explain that much, but metabolism is the process of processing all the nutrients by enzymes. They need cofactors such as vitamins in order to function properly. Of course, we need transporters to ensure that the metabolites of interest reach the proper place, and this can be the right organ, or this can be the right subcellular organelle, like a lysosome or a mitochondrion. And all of this, of course, needs to work seamlessly in order for us as an organism to be able to grow and to develop and to thrive. I think it's quite remarkable when things work well. So what happens when things go astray? Well, there are numerous genetic disorders in which an enzyme or a cofactor or a transporter is dysfunctional. There are more than 1,500 different inborn errors of metabolism that affect the ability of the body to break down or to process the nutrients, such as carbohydrates or fats or proteins. And in many of those diseases, we can see an accumulation of specific metabolites. Perhaps we can see shortages of metabolites or even aberrant metabolites appearing in the body, and those can be toxic. So what are these disorders? Well, clearly we know that we need energy for every single process in our body, right? So if there is an inborn error of metabolism, this can be life-threatening. And metabolic diseases affect many families. In the Netherlands alone, there are about 10,000 families dealing with it. As I mentioned, these are inherited disorders. There are more than 1,500 genes from which we know where a variant in that gene actually results in an inborn error of metabolism. And I should say, much to our regret, 1 in 4 patients does not reach adulthood. 1 in 4. So clearly, there is a huge need for treatment. And I think that with the increasing opportunities of genetic therapies, in addition to the existing therapies such as stem cell therapy or dietary or drug intervention, we can really be a bit more optimistic now for future patients. The symptoms of patients with inborn metabolic disorders, they are quite diverse actually, and of course they depend on the specific gene which is defective in the patient. And the symptoms can range from, for example, developmental delay, loss of skills, to exercise intolerance, reduced consciousness, epilepsy, movement disorder, liver failure, etc. They are quite diverse. So what can we do to figure out the diagnosis if a patient presents with any of these symptoms, or perhaps a combination of these symptoms? Well, as I mentioned, there are many genes that can lead to an inborn error of metabolism. So, of course, what we can do is sequence the genome. Let me see if I can move this box a little bit. We can sequence the genome of these patients and see if we can find a variant, right? Well, DNA is transcribed into RNA and then translated into proteins. Transcriptomics is used for diagnostics to some extent, but still quite limited and can be quite challenging. And proteomics actually is not really used in diagnostics as much. We do sometimes look at a single protein like an enzyme and determine its activity, but proteomics is not really used in the diagnostic practice at the moment. So what we can do, and often indeed do, is analyze the metabolome. Right? We can determine the concentrations of metabolites, and these can be the substrates of the enzyme, they can be the products of the enzyme in the various patient fluids. We often use blood of the patient, or urine, or cerebrospinal fluids. We can determine the concentrations of those metabolites, and we can try to figure out where in metabolism things go astray. So do things accumulate? Do we lack a specific metabolite? Where is the metabolic block? And I think that the metabolome paints a very clear picture of what's actually going on in the body right now. Of course, it is heavily influenced by health or disease, but it's also influenced by exogenous factors such as diet or exercise. And where the diagnostic practice in the old days used to start basically by describing a really good detailed phenotype, and then based on the phenotype, hypothesize which disease the patient might have, and then perhaps measure a biomarker to confirm the hypothesis, and then confirm it with DNA diagnostics. So nowadays, the diagnostics more and more starts with a broad genomic analysis and goes down to the clinical phenotype. So things are moving in the diagnostic worlds. In our lab, we often use metabolomics to study inborn metabolic diseases. This can be either targeted metabolomics, this can be untargeted metabolomics, that can also be flux omics. Targeted metabolomics, we use it in a hypothesis-driven way. So, for example, in diagnostics, we suspect that the patient may have disease X. So we want to check a specific biomarker, and we will have a targeted mass spec assay with which we know we can measure that biomarker. We will quantify the concentration of that metabolite. For untargeted metabolomics, we also employ this for both diagnostics and research purposes. And we use it in a hypothesis generating way. So we do not know beforehand which markers will be of interest, but we can do a broad screening and report, well, I guess not concentrations of metabolites, but provide a semi-quantitative output, right? So, for example, these features in the blood of the patient are more abundant than in the blood of our control population. And finally, fluxomics or metabolic flux analysis or stable isotope tracing. I'll be using those terms, I guess, every now and then. That's where we study the dynamics of metabolic pathways. We know that metabolism is highly dynamic. The metabolite levels are not always informative about the pathway activities. And metabolites also often engage in multiple pathways. So, for example, here we see metabolites A, B, C, and D depicted as these circles. And in the arrows are indicated the enzymes that, for example, convert A to B and B to C, etc. So let's say in metabolomics, we see that concentration of metabolite B is increased. How do we figure out what's going on? Does this mean that there is more B produced from a metabolite A? Or does it mean that there's less B being degraded to C? So does that reaction go slower? Or does the increased level of B actually stem from a completely different origin? And is the pathway going from D to B enhanced? Right, this dynamic aspect is really hard to see when doing metabolomics, and it's extremely important for a proper interpretation of what's actually going on. And I think that that's where fluxomics or flux analysis comes into play. We can trace metabolism, and we can provide nutrients to a cell. So, for example, glucose or glutamine or leucine, and use slightly altered versions so that we can trace the atoms of these nutrients throughout metabolism. So, for example, where do all the carbon atoms of glucose end up in healthy or disease states? And the enzymes in our body cannot tell the difference between these altered nutrients and the normal ones. So for this, we use stable isotopes. Stable isotopes are variants of an element with the same number of protons, but different number of neutrons. The most common carbon atom is carbon-12, which has 6 protons and 6 neutrons. Carbon-13 is a very commonly used tracer, and it has 6 protons and 7 neutrons. So it is slightly heavier than the normal, or the more abundant, I should say, carbon with the mass of 12. There's also carbon-14 having 6 protons and 8 neutrons, so even a bit heavier. And there's actually a natural abundance of these specific carbons, so they do occur naturally on Earth, where about 1% of the carbon on Earth is 13C. So in a flux analysis, we can provide this, for example, carbon-13 glucose to a cell. Glucose, as you probably know, but maybe not, has 6 carbon atoms, and we can make sure that even one carbon atom, for example, is C13 or 2, or all 6 carbon atoms are C13. We can then provide that to a cell, and the cell will convert it to, for example, pyruvate through glycolysis. And then, of course, the pyruvate will also have the 13C carbon atoms, because the enzymes won't know the difference between the carbon-12 and carbon-13. However, our mass spectrometers, of course, will, because it is slightly heavier, also the pyruvate formed, and we can clearly distinguish the labeled pyruvate from the regular pyruvate. And we can then determine what fraction of the pyruvate in the pool is actually stemming from the glucose that we gave the cells. Now, carbon-13, of course, is not the only stable isotope around. There's also, we can use stable isotopes like of hydrogen, of nitrogen, of oxygen. And there's really several advantages to these stable isotopes. Of course, well, the name stable isotope, it's very stable, so it does not spontaneously decay and release rays. So they are not radioactive. They are very safe to work with. And since they have similar properties as the natural element, it does not require any additional environmental regulation. The limitations can include the costs, of course. It's much more affordable to perform your experiment with regular nutrients rather than labeled ones. And the experiments can be quite complex because you're interpreting time-dependent data of multiple metabolic processes at the same time. So it's quite complicated to actually analyze the data, or can be at least. So how do we employ this technique in our lab? Well, I will give a few examples of how I think it may aid in diagnostics, in understanding pathophysiology, and in discovering treatment opportunities. And also, I would like to ensure that I also show you that the flux analysis can be done in vitro, in a cellular assay, for example, or ex vivo, so in patient-derived material drawn straight from the patient, or in vivo, in this case, well, fish, mice, or humans. So the first example is about understanding pathophysiology in an in vitro cell culture system. And what you see here is a graphical depiction of the carbon-13 labeled glucose tracing through glycolysis, from glucose all the way through glycolysis to the serine synthesis and into the citric acid cycle, or the TCA cycle, or Krebs cycle. And in this case, we traced cells with a deficiency in pyruvate dehydrogenase, which is located here. Pyruvate dehydrogenase is a mitochondrial matrix multi-enzyme complex, and it provides a link between the glycolysis and the Krebs cycle. Right, it is responsible for the conversion of pyruvate to acetyl-CoA. Now, when we traced the fate of glucose in pyruvate dehydrogenase deficient cells, and this is work done by Niels Meyer in our lab, we see the following. Each graph on the slide, on the left-hand side is the wild-type cells over time. On the right-hand side, pyruvate dehydrogenase deficient cells over time. And we can express the formed labeled metabolites as the total intensities or as the fractional enrichment, right? Meaning how much of, for example, the total lactate is actually formed by the labeled lactate. And the isotopologues are described as follows. So in black, M+0, meaning there is no labeled carbon atoms. M+1, meaning there's one carbon atom of the molecule labeled. M+2, there's 2 carbon atoms labeled, etc. Right, so there's a lot of data coming out from just one experiment. And we see that labeling of the glycolytic compounds, so as seen by the starting of the coloring of the bars, is already happening well within 1 minute after giving the labeled glucose, and almost all pyruvate is labeled after 24 hours. Now, based on the role of pyruvate dehydrogenase, so the conversion of pyruvate to acetyl-CoA, we anticipated that the pyruvate dehydrogenase deficient cells would accumulate pyruvate, right? Because they cannot convert it into acetyl-CoA, which would enter the Krebs cycle. And indeed, we see in the pyruvate dehydrogenase deficient cells, there's enormous amounts of pyruvate accumulating. So right in front of the metabolic block. What we also see is that there is much less acetyl-CoA formed. Well, actually, what we see is less acetyl-carnitine formed in these cells as a reflection of the level of acetyl-CoA. And also all the other Krebs cycle intermediates actually are hardly formed. You can see citric acid cycle, there's almost nothing formed in PDH deficient cells. But also all the other intermediates, you can see that, for example, the fumaric acid that is present is all black, so it does not stem from the labeled glucose. So clearly, we can use these flux experiments to get a little bit of more insight into what's happening with metabolism, and the PDH deficient cells actually behave as we would expect. They accumulate pyruvate and they have decreased Krebs cycle intermediates and also decreased acetyl-carnitine. So this was not much to our surprise, but good to see and confirm what we wanted to see. So another example of a metabolic flux where perhaps the insight obtained was not as intuitive as the former example is shown here. These are experiments performed in fibroblasts. These fibroblasts are obtained from patients with a GOT2 deficiency. GOT2 encodes the mitochondrial glutamate oxaloacetate transaminase, and is a component of the malate aspartate shuttle, as you can see here in this picture. And you will hear much more about the malate aspartate shuttle in Hanna's presentation. The only thing you need to know now is that it is important to get NADH into the mitochondria. So patients with a GOT2 deficiency are expected to have a disturbed NAD-NADH balance, and patients present with a metabolic encephalopathy and epilepsy. What I wanted to show here was that indeed patients with a GOT2 deficiency indeed show a disturbed NAD-NADH balance. But we know that NAD and NADH are important for so many more metabolic processes. And when we look at glycolysis, for example, we've performed glucose tracing in these patients' fibroblasts. Of course, we see from glucose, we go through glycolysis, we make 3-phosphoglycerate. 3-phosphoglycerate, however, cannot just proceed through glycolysis. It can also be formed in multiple enzymatic steps into serine, right? And by doing flux analysis, one process that we saw being severely hampered in these GOT2 deficient patient cells is the synthesis of serine from glucose. And serine is very important for our neurodevelopment. It can act as a neurotransmitter, and we know that low serine due to other genetic defects can result in epilepsy and encephalopathy. And importantly, we also know that serine can be given to patients as treatment, right? So these GOT2 patients were indeed treated with serine supplementation, and they show a very good response upon supplementation when looking at the epilepsy. Now, I have to say that this was a combination of serine and vitamin B6, so it's hard to figure out, of course, the role of each individual component, but we do know that the serine supplementation can be very beneficial for patients with a disturbance of the serine production. So in this way, I think we can also learn from the stable isotope tracing whether there are components that that we can supplement back to the patient and perhaps alleviate the severity of disease to some extent. Well, doing flux analyses in cultured cells is quite complex, but still far easier than in other systems and in bigger systems. but it does teach us a lot about pathophysiology and biomarkers. But I wanted to briefly mention that it can also be performed in, well, for example, patient-derived liver organoids, so a 3D model. In this case, the organoids were derived from the liver of a patient with methylmalonic acidemia, or MMA, metabolic disease. Patient will have mutations in the enzyme right here, involved in the breakdown of valine. The patient is expected to accumulate methylmalonic acid, hence the name methylmalonic acidemia, I guess. And of course, for this particular disease, we cannot trace glucose, or we could trace glucose. Not sure what it would tell us. But what we did is we traced labeled valine and looked at where in metabolism things started to be different from our control situation. So these were liver organoids from the patient and from control. And we saw actually not super unexpectedly that propionyl carnitine and methylmalonic acid in the patient, so right upstream of the block, started to accumulate in these 3D liver models. So now we can go on and study other alterations in these patient organoids in more detail, and perhaps even use this model as to test effectiveness of different treatments. So that's the in vitro work I'll show, and Hanna will show some more cell culture work, but I also wanted to highlight what can be done in fresh patient material, specifically in blood drawn from the patients, ex vivo analyses. In this case, red blood cells. Well, as you know, red blood cells are quite different from most cells. Mature red blood cells have no DNA, no RNA, nor organelles. And their main function is to transport oxygen, which is also well, represented by the proteome, which is largely in favor of just doing this function. They say metabolism of the red blood cell is relatively simple, although I think I dare to say that it never really is. When we look at the glucose metabolism in the red cell, we have what we call the Embden-Meyerhof pathway, which is basically glycolysis of the red blood cell. So it's producing energy for the cell, from glucose all the way, of course, to pyruvate. There is the pentose phosphate pathway, which mainly functions in the red blood cell to reduce the reactive oxygen species. And there's the Rappaport-Lubering shunt, making 2,3-bisphosphoglycerate, which binds hemoglobin and regulates oxygen affinity. So what we did is we draw blood from the patient and isolated red blood cells, immediately traced the fate of glucose in the blood of that patient. And I'm not going to tell you yet what the diagnosis of the patient is, because I think you can figure it out based on our Flux results. And don't worry if you don't, because I will tell you in the end what the diagnosis is. So first we look at the upper glycolysis part, from glucose to glyceraldehyde 3-phosphate. On the left-hand side, healthy controls, the glucose flux up to 6 hours, and then the patient 0 minutes up to 6 hours. And for the upper glycolysis, so glucose 6-phosphate, fructose 6-phosphate, glyceraldehyde 3-phosphate, we do not really see a clear difference in the label form, so the blue part of the bar, or in the total levels of these metabolites. So I think that the defect in this patient is probably not in this part of metabolism. When we then move to the pentose phosphate pathway, we start to see things happening, things being different in the patient compared to controls. Not so much here, but when we look at cetoheptadose 7-phosphate, it's right over here, we see that the patient here, the right half of this graph, accumulates a lot of cetoheptadose 7-phosphate, not necessarily directly formed from the glucose we provided, some of it is, but the levels of cetoheptadose 7-phosphate are really quite strongly elevated. So perhaps now you know the diagnosis. I wouldn't know for sure yet based on these data, but this actually makes things a bit more clear, I think. When we look at the lower part of glycolysis, so from bisphosphoglycerate down to phosphoenol pyruvate, we see in the patient red blood cells that all these metabolites accumulate, and also they are formed from, in blue, from the glucose we provided. When you look at phosphoenol pyruvate, it's really quite high compared to our healthy control red blood cells. So perhaps when all of this is accumulating, perhaps there's a block below. So we look at pyruvate and lactate. And indeed, in the patient red blood cells, there's almost no pyruvate, here in blue, almost no pyruvate formed from the glucose, meaning that there's most likely a block between phosphoenol pyruvate and pyruvate. And then I think it is relatively easy to figure out that this patient most likely has pyruvate kinase deficiency. And that's indeed the diagnosis of this particular patient. So we can do ex vivo flux analysis, in this case for a pyruvate kinase deficient patient. We can trace glucose metabolism in red blood cells. The pyruvate kinase deficient patient shows a decreased production of pyruvate and lactate, and accumulation of the glycolysis intermediates and the pentose phosphate pathway intermediates upstream of pyruvate kinase. And we can pinpoint the level of the defect based on the stalled flux. And I think this may have implications for diagnostics, for understanding pathophysiology, and for treatment. And I say treatment because we know that there are drugs developed that alter the enzyme activities in these pathways for the patients. And in addition to pyruvate kinase deficiency, we're also studying fluxes in other red blood cell disorders, which I think now show really exciting results. Finally, the last slide, I cannot talk about flux without telling you that it can also be done in vivo. We do not do this in our lab, but I can refer you to some beautiful papers which are published. For example, this one, where the authors have really done some important work and really a lot of work, where they supplied mice with 15 different nutrient tracers. And they followed the fate of all of those compounds in the entire mouse. Fed the mice different types of diet and see how that alters metabolism. So I think this is a very powerful tool to get really very in-depth information of alterations of metabolism. So before I give the floor, Hannah, to you, I would like to acknowledge the people who are involved in this work. The people of our metabolic diagnostics lab. I mentioned work of Niels, Hannah, Titina, but also the red blood cell work is done in close collaboration with the red blood cell research group here. I would like to thank all the contributors, and I am looking forward to your questions after Hannah's presentation.
So thank you so much, Judith, for a really interesting presentation. And we already have some questions piling up. Please, if you have some more questions, just pop them into the Q&A panel. But we will now continue with Hannah's presentation, another lecture, and then we will wrap up with the Q&A session.
Yes, I will just share my screen. Okay, I hope you can all see the presentation and hear me well.
Perfect. It's
Yeah,
perfect.
all good.
Okay, so my name is Hannah German. I'm a PhD student at the Metabolic Diagnostics lab of Judith Jans, and today I will talk about our project on studying malate aspartate shuttle deficiencies and how we use stable isotope fluxomics to elucidate the pathophysiology of these deficiencies. So to introduce a bit, the malate aspartate shuttle, or MAS for short, is a shuttle that spans across the inner mitochondrial membrane, and its most important function is to shuttle electron carriers across this membrane, because the membrane is impermeable to NADH. This carrier system is necessary to transport it across. So here you see kind of a schematic depiction of the shuttle. It consists of 4 proteins and 2 carriers. In the mitochondria, we have malate dehydrogenase 2 and GOT2, and then in the cytosol, we have their cytosolic equivalents, malate dehydrogenase 1 and GOT1. And then we have the 2 carriers, the oxoglutarate carrier here on the left and the aspartate glutamate carrier on the right. They shuttle these metabolites across the membrane, and basically the net result of this is that malate dehydrogenase 2 in the mitochondrion produces NADH, and then malate dehydrogenase 1 in the cytosol recycles NAD+. And also the shuttle is responsible for the production of aspartate in the mitochondria and transporting it out into the cytosol, which is important for example, the urea cycle. And it also produces glutamate. So if we zoom out a bit, we can see the entire central carbon metabolism, and we see that the malate aspartate shuttle, which is here on the left, is closely related to the citric acid cycle, with NDH2 here also being part of the citric acid cycle. The production of NADH is, of course, very important inside the mitochondria for the electron transport chain to produce ATP. And the production of NAD+ in a cytosol by MDH1 is important to drive several cytosolic processes, such as glycolysis, which is NAD dependent, but also the biosynthesis of serine, which Judith already introduced a bit. And this is also the first step is NAD dependent. And as you can also see in this figure, there are other pathways in the cytosol that can produce NAD, such as the activity of lactate dehydrogenase and also the glycerol 3-phosphate shuttle. But NDH1 is mostly the most important source of NAD+ in the cytosol. So we study deficiencies of the malate aspartate shuttle, and these are cases where there's a pathogenic mutation in the gene leading to deficiency of the enzyme. And as you can maybe imagine, these are ultra rare because the MAS is such an important process. Deficiencies are currently known in literature for both malate dehydrogenases, for GOT2, and for both isoforms of the aspartate glutamate carrier. So we have the neuronal isoform, which is AGC1 or RLR, and then we have the hepatic isoform, which is AGC2 or citrin. And yeah, so there, as you can see in the second column, these deficiencies are very rare. They're usually only a few patients known, except for citrin deficiency, which is a little bit more prevalent. And the clinical presentation for most proteins, which are mainly expressed in the brain due to their importance in ATP production, is then a neurological phenotype. So we see epilepsy, developmental delay, microcephaly, dysmorphic features, hypotonia. So really a neurological phenotype, whereas for a citrin deficiency, it is more a hepatic phenotype, which is to be expected. And then the biochemical findings range quite a bit, but what we can see is that it's often accompanied with high lactate levels, high glycerol 3-phosphate levels, sometimes high ammonia. And this again makes sense as lactate and glycerol 3-phosphate are products of NAD regenerating processes, like I just said. So this is likely a compensation mechanism for dysfunctional malate aspartate shuttle. So how do we use fluxomics to study these deficiencies? We have different 13C labeled nutrients, like you just introduced, and the main metabolic sources for the citric acid cycle and the malate aspartate shuttle are glucose, glutamine, and also fatty acids. So we use 13C labeled versions of these metabolites in combination with cell models. And the main cell models that we use are either fibroblasts derived from patients. But as you can imagine, as these diseases are very rare, it's also quite difficult to get patient-derived models. So we don't have many of these, but we have a few. And then we also created a panel of knockout cell models of each different MAS enzyme in HEC293 T cells. So we also use these a lot in combination with these labeled nutrients to study the pathophysiology of these deficiencies. So we incubate these cells with these labeled nutrients for a set amount of time or for several time points. And then we analyze them with mass spectrometry. And we either use targeted approaches to, for example, look at intermediates of the TCA cycle, glycolysis, the pentose phosphate pathway, and we can also measure NAD and NADH. Or we can use a more untargeted approach based on direct infusion mass spectrometry, which yields us about 70,000 different MZ values. And we use this in combination with a library of labeled metabolites to look at labeling in a more hypothesis-generating manner. And our main goals with this method is to elucidate the pathophysiology of MAS deficiency, so look more closely at what goes wrong in metabolism when these enzymes are deficient. And this, of course, goes hand in hand with gaining a deeper understanding of the function of the malate aspartate shuttle and how exactly it works. And then lastly, we also use it to discover therapeutic mechanisms. Because these diseases are so rare, there are no targeted therapeutics currently available, and most patients are treated with anti-epileptic drugs and a ketogenic diet, for example. So I will give you an example of some studies that we did. So first, a study that we did to look further into the pathophysiology and the functioning of the MAS. So like Judith already mentioned, in GOT2 deficient, in one of the GOT2 deficient patients, there was low plasma serine observed, and it was found that the seizures of several patients were responsive to serine. And the hypothesis that was generated from this is that the biosynthesis of serine is hampered in these patients because there are low NAD levels. And like I just mentioned, NAD is necessary for serine biosynthesis. So serine is synthesized de novo from 3-phosphoglycerate, which is a glycolytic intermediate. So that makes it so that if we add 13C6 glucose to the cell, this will yield 13C3 labeled serine, which you can see here in this scheme. So we can actually follow serine biosynthesis over time by adding labeled glucose in fibroblasts of GOT2 deficient patients. And in this way, we can see whether there's a block in serine biosynthesis. And this was found in a study in 2019 to be the case. So in this plot, you can see the biosynthesis of labeled serine over time in GOT2 patient fibroblasts, which are here in the dashed lines. And you can clearly see that this is decreased compared to the controls and the carriers, which are in the solid lines. It's still a bit higher than these bottom two dashed lines, which are patients that have deficiencies in serine biosynthesis enzymes, so BSET and 3-BGDH. So using this flux analysis, they could actually see that there is indeed a serine biosynthesis block in these patients. So then we hypothesized that if we add pyruvate to these cells, which can regenerate NAD+ to form lactate, that then this serine biosynthesis might be rescued if it is indeed NAD+ dependent. And this was indeed the case. So in this scheme, you can see, again, the fibroblasts that we incubated with different concentrations of pyruvate. And here we can see that with 0 millimolars of pyruvate, serine is decreased in the patient cells compared to the carriers. And then if we add pyruvate, it is rescued. And this is also the case for glycine, which is the downstream product of serine. So this tells us that indeed the serine biosynthesis block in these GOT2 deficient patients is NAD dependent and can be rescued by regenerating NAD. And then, of course, we can ask ourselves, do then all malate aspartate shuttle defects lead to this secondary serine biosynthesis defect? Because the lack of NAD is not a GOT2 specific effect, but of course, an effect of dysfunction of the malate aspartate shuttle overall. And this was indeed the case. So this is a project done by Melissa Brooks, a PhD student from our lab. And here we used our panel of HEC293T knockout cells of each MAS enzyme. And what you can see here in the plot on the left is the wild-type cells in black. Again, the serine biosynthesis over time. And we can see that there are varying degrees of serine biosynthesis impairment in the different knockout cells, with MDH2 knockout being the least affected and the MDH1 knockout being the most affected. And then here we can also see the advantage of doing a flux analysis, because if we look at total unlabeled serine levels in these cells, which is the plot on the right, we can see that there's no significant difference, and in some cases it even seems slightly increased. So using the 13C label, we can actually distinguish the de novo biosynthesis versus serine that is derived from other sources, such as taking up from the medium. And we can actually see that there is indeed a defect here. And then we also showed that this serine biosynthesis block is accompanied by an increased lactate pyruvate ratio in these cells, and also a decreased NAD/NADH ratio. And we could see a nice inverse correlation. So the cells that had the least serine also had the highest lactate pyruvate ratio, and vice versa, which is even more evidence that it is indeed a redox-dependent effect. So now another project that we did to discover therapeutic mechanisms in MAS-deficient cells. And now we will talk a bit about deficiency of malate dehydrogenase 2, which I already mentioned is the enzyme that connects the malate aspartate shuttle to the citric acid cycle, and it's also the enzyme that produces NADH, so a very crucial enzyme in both processes. And as you may expect, cells that are deficient in MDH2 have a significantly decreased TCA cycle activity. And that is when it was suggested that anaplerotic therapy could be a valuable strategy for these patients. And maybe you know the term, maybe you don't, but anaplerosis is the replenishment of TCA cycle intermediates. And this can be done through different substrates, but one of them is triheptanoin, which you can see here in the scheme on the left. And this is a triglyceride made up of 3 C7 fatty acids, and these can be metabolized through fatty acid oxidation to acetyl-CoA and succinyl-CoA. Because it has an odd number of carbons, it will lead to these two products, which can both be entered into the citric acid cycle at different points. So it is hypothesized that this can then replenish the TCA cycle in these MDH2 deficient cells. And it has also been shown already in one MDH2 deficient patient that this supplementation of this triheptanoin led to relief of the symptoms. But the metabolic mechanism through which this occurs has not yet been elucidated. So what we were wondering is, do these fatty acids contribute directly to the TCA cycle? So do they really contribute carbons, or does it work via an alternative mechanism? So how we study this is we used again our MDH2 knockout HEC293 T cells that we already had, and we incubated them with 13C3 C7, which you can see here on the bottom, and also 13C2 C8. C8 is a fatty acid with an even number of carbons, so this can only lead to acetyl-CoA, not the propionyl-CoA. So we wondered, does this make a difference if the fatty acid can provide both substrates or just acetyl-CoA? So you can see here in the scheme how these metabolites are entered into the citric acid cycle. So they both undergo fatty acid oxidation. And it is important to note here that because only the last carbons are labeled, the C7 will only lead to labeled propionyl-CoA, and the C8 will only lead to one labeled acetyl-CoA. And then again, we can use mass spectrometry. And because these have labeled carbons, we can see a nice mass shift. So we can actually determine if the carbons of the citric acid cycle are directly derived from one of these substrates. So first we checked if these fatty acids are metabolized through fatty acid oxidation. So first for heptanoate or C7. And what we can see here nicely is that if we add it to our wild type or knockout hex cells, that the acyl carnitines that follow, so the C7, C5, and C3 acyl carnitines are nicely labeled. So it is here plotted as the fraction of the total pool that is labeled, like Judith already explained. So we can see here that the majority of the pool of the acyl carnitine becomes labeled after 8 hours of incubation with the labeled fatty acid. So this indicates to us that it is efficiently metabolized through fatty acid oxidation in both our wild type and our MDH2 knockout cells. And then we can also check this for C8. And here we see a little bit less efficient labeling for the knockout cells of the acyl carnitines. So these are then even chain acyl carnitines. But still, we see labeling. And then for acetylcarnitine, we see a markedly lower fraction of labeling, which makes sense because these experiments were done in the presence of glucose. And we know that glucose is the main source for acetyl-CoA. So it makes sense that only a small fraction is derived from fatty acids. But we don't see a lot of difference here between the wild type and knockout cells. So this indicates that the label does end up in acetyl-CoA efficiently. So then we look at citric acid cycle intermediates, and we can see here for C7, the fractions of labeled succinate, which is where it enters, and malate. And we did this experiment for cells that were untreated, cells that were treated with unlabeled C7, and cells that were treated with labeled C7. And what we can see is that the cells that were untreated or with unlabeled C7 do not show significant labeling, which is what we expect. So that is a good check that we are not looking at noise. And then for the cells where we add the labeled C7, we see a nice enrichment of 13C3 in succinate and in malate, again for both wild type and MDH2 knockout cells. And the labeling enrichment does seem a bit less in the knockout cells, which is also expected because they had less TCA cycle flux. But it's still nice to see that they are able to incorporate the label into these metabolites. For C8, we do not see this, so we do not see increased labeling for the knockout cells, while we do see it for wild type cells. So acetyl-CoA enters via citrate, and we see nice citrate enrichment in the wild type, but not in the knockout. And if we look at the scheme here, we can maybe also expect that because MDH2 is responsible for making oxaloacetate, which is the acceptor of acetyl-CoA to form citrate. So if MDH2 is deficient, there is likely not enough oxaloacetate available to form citrate. And we can also look at amino acids, which are derived from the citric acid cycle by the malate aspartate shuttle, which is glutamate and aspartate. And here we actually see the same results that we saw for the citric acid cycle. We see that glutamate and aspartate are nicely formed from C7, but not from C8 in the knockout cells. So this tells us that indeed the odd chain fatty acid is a more efficient anabolic substrate in these cells than the even chain carbon fatty acid. Then we also looked at the redox balance, because as you can maybe see on this previous slide, if we look at the y-axis, the fraction of the total pool of these metabolites that are labeled is actually quite small. So this cannot, we figured this cannot account for the entire picture. So we looked a bit further into indirect effects. So here we see the total levels of NAD+ and NADH and also the ratio of these two. And what I hope you can appreciate is that in the untreated knockout cells, there is a decreased NAD+, NAD+ ratio, which is what we expect. And if we add C7 or C8, that this is increased. And C8 is even able to normalize the NAD-NADH ratio above the wild type level. So this indicates that the fatty acids do not only contribute carbon to the citric acid cycle, which is how anaplerosis works, but also indirectly affect the redox ratio in the cells, which is very nice to see. And then we again also looked at the serine biosynthesis, which we know is deficient in these cells, and also the lactate pyruvate ratio, which we know was increased. And we again see a nice normalization of both of these. So this again indicates to us that the main mechanism of these fatty acids, at least in this cell model, is correcting the redox balance. So that brings me to my conclusions. I hope I've showed you that Fluxomics is a valuable tool to study metabolic diseases. We can trace metabolites from a specific source. We can distinguish different pathways, and it gives us higher resolution. We can use it to understand disease pathophysiology because we, for example, showed that deficiencies of the malate aspartate shuttle lead to a secondary serine biosynthesis defect. We can nicely correlate this with the NAD-NADH balance. But we do recognize that there's a need for more patient-specific models. So we now use HEC293 knockout cells, which are nice models, but in practice, patients never have a full knockout of the enzyme. They have one mutation with usually some residual activity of the enzyme. So it would be nice to get some models that are more specific to the patient background. And then we can also use it to further elucidate mechanisms of therapeutics, such as the anabolic mechanism of medium-chain fatty acids. And we could nicely show the redox rescue by pyruvate in the GOT2 cells and by the medium-chain fatty acids in MDH2 cells. And this then again prompts further research into the exact redox regulating mechanisms of these therapeutics. So with that, I would like to thank everyone from my group who helped on this project. Also from the UMC Groningen, Barbara and Albert, who helped with the MDH2 project. Fried and Denise, who did a lot of work on making the knockout cell lines. And Reza from University College London, who spearheaded the GOT2 project. And I would like to thank you for your attention, and I'm happy to take any questions.
Okay, thank you so much. Very, very fascinating talk, and it's really evident how complex it is. What are all the mechanisms happening in cells, and you show with your data how you can really decipher it piece by piece.
Yeah,
fascinating. Yeah, I have a lot of questions here, but there we have a lot of questions in the Q&A, so we probably will jump right into them. So one of my questions, and it's also kind of reflected by other questions in the Q&A from the attendees, is because you showed a lot of data, of course, and there had to be a lot of data processing and maybe some bioinformatics. So I would like to ask you, maybe both, whoever answers, what software do you use for the also, of course, annotation, but also this isotope ratio, and generally, how do you decipher this data?
Shall I start, Hannah? And then just please fill in if you want to add. So I guess this differs a lot from, it depends on whether we do untargeted approaches or targeted approaches, right? For untargeted approaches, we indeed have in-house developed pipelines that do the peak calling, and the annotation is then, as I saw, one of the questions was raised, the peak annotation is then based on the human metabolome database. However, that is, of course, when we use direct infusion mass spectrometry, that annotation is putative, and then we would need to confirm with a targeted assay. And for that, we have dedicated assays specifically developed to be able to quantify those metabolites. And I don't actually remember what software we use for that, but that's commonly available, I think, Hannah.
For the
targeted analysis.
yeah, we use it's from Thermo Fisher. It's called Trace Finder, and yeah, it comes with the mass spectrometer, I think. So yeah, it's a nice.
Okay.
But yeah, indeed, I can add that I think the models that we have where we can expect certain metabolic effects, such as the pyruvate kinase deficiency or the pyruvate dehydrogenase deficiency, that we can nicely use those models to validate the labeled metabolites that we are looking for. So, for example, if we expect an accumulation of labeled pyruvate, that we can nicely check if we are looking at the right mass. Yeah.
Okay, so it's mainly commercially available or free-to-use software, or do you also develop something by yourselves?
Yeah, so we have the in-house pipeline. It's in R, and
Okay.
yeah, that's mostly for the untargeted data.
And
is
it available in your papers, maybe?
Yes.
Like a link?
Yes.
Okay,
Yeah.
perfect. So if anybody is interested, you can link to your
Yes,
papers
it's
and
on
find it there.
GitHub. So yeah.
Perfect. Thanks a lot. There are also a lot of questions about the methodology. For example, if people want to adopt your methodology, or what are some tips or tricks for tracer metabolomics. For example, one of them was focused on the timing. What time points to use where doing tracer metabolomics? Do you have any tips for that, maybe?
Yeah, I think it's mostly trial and error, but it's also, I think, partly knowing the cell model or model that you're working with, and how fast they metabolize certain nutrients. For example, in the HEC cells that we use, they metabolize glutamine very fast. So you would have to work with probably something like 10 minutes, 30 minutes, such time points, whereas glucose is a bit slower. So yeah, I think it's mostly trial and error, and doing some research on the cell model that you want to investigate.
So, for example, the fluxes that you showed on the serine flux from glucose in the fibroblasts of the patient, it's much slower than in the HEC cells. So it depends also on the cell type and on the model.
Yeah, sure.
Okay, so then you also mentioned the organoids that you're working with. It's quite a hot topic now, I would say. And so I was wondering, and also there was one of the questions there from the attendees. How do you perform tracer studies in organoids? Is there some very different specific compared to cells, or?
Yeah, so yes. I think I find them quite complicated, but it's hard to normalize your input material. So we did some organoid stuff a couple of years ago when when the technology was not as developed yet, and then we really had a hard time getting also measuring the metabolites appropriately was really difficult. So now what we did was we transferred the liver organoids, and these were I think intrahepatic cholangiocyte organoids. When they we need quite a bit of material compared to more normal organoid work. So we then transferred the 3D organoids for some time in liquid medium. And you can also do that in spinner flasks, I think, nowadays, to increase the yield a little bit. But so we transported them to a liquid medium, because usually they grow in the matrix, in the gel. But the gel interferes too much. So we moved them to liquid, kept them there for a short time, and then added the isotope tracers. I agree. It's more tricky than just a 2D culture where you can wash away all the supernatants and just add whatever you want. It's not as easy, but I think it's nice to have the combination of the cells in there. Yeah.
Yeah. And as you also mentioned the troubles with the normalization, did you figure a way how to normalize these data from organoids, or?
No, well, in that sense that I wouldn't. So what we did first was try direct infusion mass spectrometry, comparing that does not work properly when you just want to quantify metabolites. So when you do it in a targeted way, you can look at ratios of different metabolites, And that tells you something. Well, if you do it quantitatively, then at least you can say a bit more about about how to normalize.
Okay, thanks a lot. So we have quite some more questions. I hope we have some more time, maybe 10, 15 minutes if it's fine. Just stop us if it's enough, and people then can reach you by email, of course. And I will pass to Loic for some of the next questions.
Yes, thanks, Aleš. So I can continue with some questions in the chat, but also I have my own questions as well. But first, I would like to thank you both for these talks. We have questions for Anna from Mickey Alster. So the first one is, in the experiment of the knockout, he was wondering if you saw some changes in the glycolytic rate, as many steps of the glycolysis are NAD+ dependent.
Yeah, so in the fibroblasts, I think you mean?
Yeah.
Yeah, it's I didn't show it, but we also we looked extensively into glycolysis in these cells, and basically the most efficient cells all have blocked glycolysis. So we see indeed what you already mentioned, the and above the NAD+ dependent step of GAPDH, we see accumulation of glycolytic intermediates, and then below the NAD+ dependent step, we see decreased glycolytic intermediate. So it's clear that the NAD+ dependent step of glycolysis does not work properly in these cells. And then, of course, you could also ask yourself, is that then the reason that the serine biosynthesis is deficient? But we actually also looked at the ratio of phosphoglycerate and serine, and this was still decreased. So it is really two separate occurrences.
Thanks. And also talking about serine, I had a question about this. So you show nicely that you have a decrease in serine synthesis, but not a decrease in the serine total pool. And I think that's an interesting feature in itself.
Yeah.
Because does it mean that you use less serine, so maybe less serine entering the 1-carbon metabolism, and the cells is just preserving the serine pool? So how would you explain that?
Yeah, we are actually looking into that as well, because we thought it was striking that even sometimes we see increased serine levels, which is very surprising. And one hypothesis that we have, especially for the cell models, is that they just take up more serine from the medium because it's in the medium.
Have you checked that?
Yeah, and it's maybe a little bit more, but not, I don't think it would explain it entirely. So we are indeed looking now into 1-carbon metabolism, if there's maybe a change there indeed in the use of serine. Because yeah, it's something that we didn't expect to see. Yeah, that the serine levels would be normal even if the biosynthesis is so much decreased. Yeah.
Thanks. One more question about your studies, and someone was wondering for the fatty acid labeling experiment, if there was a particular consideration why you used C13 2-carbon labeled octanoate rather than the uniformly labeled. Is it because of like availability of standard, or do you have a real purpose by doing C2?
Yeah, so what our consideration was is that we already had the 13C3 heptanoate, which I think was the only one available at the time, but I'm not sure. And this, so this was the last 3 carbons of the fatty acid which were labeled. So this is going to be the carbons that go into propionyl succinyl-CoA. So to make an equivalent comparison between the two, we figured that we would need the octanoate, which also makes 1 acetyl-CoA. Otherwise, the comparison would not be equal. If we would get 4 labeled acetyl-CoA versus 1 labeled propionyl-CoA, then you cannot really compare which one contributes more or less to the citric acid cycle. So that was our consideration, to use the octanoate that only produces 1 labeled acetyl-CoA. But I think the uniformly labeled is also available, so it is possible to use that one as well.
Thanks. Now we have more, again, methodology question, and asking how you correct for natural isotopic abundance. So do you use like an R script already made?
Yeah, so we usually, like I already showed, we take along samples that were not labeled. So then we we can check if we have noise labeling basically in the samples that are not supposed to have label. And that is basically our correction for natural abundance. But I do know that there are also groups that really use like mathematical equations to correct for natural abundance. Yeah, but we don't do that, so I cannot say too much about that. But
okay. Now I had a question for Judith mostly. You showed in your initial slides an example of metabolite A, B, C, and D, and say that without isotopic tracing, it's hard to tell that if it's a defect in the enzyme consuming the metabolite or producing the metabolite, and I completely agree with that. Do you think that proteomics, that you said it's not widely used in diagnostic, could be a good add-on, as if you detect something happening in one proteins that is producing one metabolite, that could give you a hint of maybe what's happening? And do you think that it's something that can be doable?
Yeah, I think so. It's not commonly used in diagnostics. However, I think when you are investigating a model system of a particular disease, then you can use metabolomics, proteomics. Sorry, proteomics. I think is really hard to do on an individual patient, and that's the main problem. That it's difficult to compare proteomes between human subjects because they vary a lot, and but I think proteomics can definitely aid in when, for example, the models that Hannah described, where you have knockout and patient models and control models, where you then see metabolite accumulating, and you can check whether the protein of downstream the metabolite, for example, is higher or lower. Yes, I think definitely proteomics is very important, but not yet ready to be used in clinical diagnostic practice, I would say.
Thank you. And we had a question about what were the pros and cons of using direct infusion instead of LC when performing isotopic metabolomics? So do you have like an opinion on that?
Yeah, so I think so I showed some work of Niels Meyer from our lab, and he used direct infusion, actually, so an untargeted way to look at metabolic fluxes. The advantages there are that you can have a very broad view of what's going on in metabolism. The disadvantages are clearly that the identifications are putative, right? You need to confirm with a targeted LC-MS method, because otherwise all the metabolites with the same mass cannot be distinguished. So I think similarly to our diagnostic approaches, that the direct infusion methods are very suitable to in a hypothesis generating way, whereas confirmation would most likely need to come from LC-MS methods with a targeted approach.
Thank you. And would you have any recommendation in terms of minimum samples number you can use for studies, like in terms of cell culture, animal or clinical studies, like a minimum that would provide good quality and good data?
Do you mean so the minimum amount of cells or the minimum number of different samples?
I guess the question was mostly about the minimum number of different, I mean, the N per condition, basically.
Yeah, Hannah, I think we mostly use NS3 samples for all experiments where we have biological triplicates, so not just a technical mass spec triplicate, but biological triplicates.
Yeah, sometimes for patient material it is difficult because you sometimes limited to how much material you have, but especially with cells, yeah, we always do triplicates. Yeah.
Thank you. I think that's it for me. I'll give the mic back to Aleš for if he has any additional questions, maybe.
Yeah. I think there are just like few more, so I will just ask these if it's fine for you. I think this one, I was wondering if you were concerned about potential isotope scrambling with the long labeling times of 24 hours.
Yeah, I imagine 24 hours is quite long indeed, and I can imagine that there's some scrambling. So that's why, like Hannah explained, you really need to know your system, and you need to check different time points to make sure that you're catching the processes at the right time, and not when there's an equilibrium of all the labeled atoms throughout metabolism. So in these cases, not worried, but it's definitely something to be considering.
Okay. Yeah. Thank you. And there's one more question about the separation. So if what did you use to separate the ribulose 5-phosphate from the ribose 5-phosphate? And if you see any interconversion in these metabolites in the extracts?
Interconversion, I don't know. So the method we use is published by Waters in 21, I think, and it's a Hillick method. I am not sure about interconversion, actually, whether we see that. No,
But for the separation, you use some method that really nicely separates all these.
Yeah, it's a UPLC
phosphates.
method with a Hillick column that we use for separation. Indeed, yeah, it's published not by our group, but from a Waters company, I think, in 2021.
Okay, we have only last 3 questions. I will run through them, if it's fine, and then we can wrap up. So one is asking about standards, internal standards, if you use any. Basically, that's the question.
Yeah, so and whether they are the same as the label that we trace. Well, of course, you have to be careful that the compounds that are made through metabolism from the tracer are not the same as the internal standards that you use. So, for example, when we use carbon-13 labeling, we can use deuterated internal standards so that we can separate the internal standard from what's actually made through metabolism. Yeah, it's a good point.
Then there is one question for Hannah also. If you did some transcriptomics also, or if you have the transcriptomics data for the, I think this is concerning the malate aspartate deficient samples.
No, we didn't. We did not do transcriptomics. We basically did the metabolomics data. We have tracer data, and also we have Western blots of the most enzymes, and also some other enzymes, but no transcriptomics. No.
Yeah, but maybe not only for this study, but you also for some other studies have combined different omics with your tracer data, proteomics or transcriptomics.
We do. We have a project currently where we did some Silec proteomics. So that's and we are trying to integrate that with metabolomics data, so to really see changes in protein expression that we can correlate with the Flux data. And it gets quite complicated because, of course, tracing data is already quite complicated, and then to integrate it with the different omics. But it's very informative, and I think it's definitely the future to do multi-omics and integrate all this data. So
Perfect. Thanks a lot. So we have some more questions, but I think for the sake of time, and you already spent and gave us a lot of your time. So thank you so much for it. It was fascinating lectures, a lot of questions, nice discussion. I'm really happy, and thank you once again. This webinar was recorded, so it will be soon available on the Metabolomic Society website and also YouTube, also for those who didn't join us today. But I would like to once again thank you so much for your great talks, and hopefully see you soon somewhere at the conference maybe, or somewhere. Looking forward to it. So thank you so much.
Thank you.
Yes, thank you.
Thank you. Have a great day. Bye.